Here are 3 high-yield USMLE-style questions focused on the genetic terms epistasis, aneuploidy, and dominant negative mutation. Question 1. A 12-year-old boy presents with albinism, and genetic testing reveals that he has a mutation in the tyrosinase gene. However, his phenotype also appears to be influenced by a second gene that alters his skin and hair pigmentation. Which of the following best describes this interaction between the two genes? A. Epistasis, B. Pleiotropy, C. Incomplete dominance, D. Codominance, E. Anticipation. Answer. Explanation. Epistasis occurs when one gene affects the phenotypic expression of another gene. In this case, albinism results from a mutation in the tyrosinase gene, but the expression of the phenotype may also be modified by the effects of a second gene. This interaction between genes, where one gene masks the expression of another, is an example of epistasis. A 45-year-old woman with a history of early-onset menopause undergoes genetic testing, revealing a 45XO karyotype, which is characteristic of Turner syndrome. Which of the following best describes the genetic abnormality present in this patient? A. Epistasis, B. Aneuploidy, C. Genetic imprinting, D. Dominant negative mutation, E. Point mutation. Answer. B. Aneuploidy. Explanation. Aneuploidy refers to the presence of an abnormal number of chromosomes due to non-disjunction during mitosis or meiosis. Turner syndrome is caused by monosomy X, 45XO, meaning the individual has only one X chromosome instead of two, which is an example of aneuploidy. Question 3. A patient with colorectal cancer is found to have a mutation in the p53 gene. The mutation causes the production of a non-functional p53 protein that binds to the wild-type p53, preventing its function. Which of the following terms best describes this type of mutation? A. Epistasis, B. Dominant negative mutation, C. Loss of heterozygosity, D. Gene silencing, E. Gain of function mutation. Answer. B. Dominant negative mutation. Explanation. A dominant negative mutation occurs when a mutated allele produces a non-functional protein that not only fails to perform its normal function, but also interferes with the normal gene product. In this case, the mutant p53 protein blocks the wild-type p53 from functioning, which is characteristic of a dominant negative mutation.